Welcome to another histology guided tour. Today we're going to look at the eye. This recording is intended as a replacement for the histology lab session. In this section, we're going to look at the microscopic anatomy of both the anterior segment and the posterior segment of the eye. This will be a fairly short lab session as there's only a couple of slides. Let's start with the 20x whole eye specimen. This specimen really doesn't have much morphological detail up at 40x because you just can't get past 20. It was scanned lower. But it does allow us to see some of the overview features and a couple of structures that aren't in some of the other specimens that we've got. So first, let's look at things like the chambers of the eye. Let's review those. The chambers are three. First, you have the anterior chamber, which is behind the cornea but in front of the iris. Then you have the posterior chamber, which is behind the iris but in front of the lens. And it's bounded by these zonules here that hold the lens in place at the sides and the lens here. So of course there is interchange of aqueous humor between this posterior chamber and anterior chamber via the pupillary opening. The third chamber is out here. It's this large vitreous body chamber, and that is usually filled with the vitreous body, or as it's sometimes called, vitreous humor. Now let's take a look at some of the tunics of the eye. They are three as well, and the outer tunic is the sclera out here, and in the front of the eye, that would be the cornea. So cornea and sclera, which is the white of the eye, that's the outermost. The next is what we call the uveal tract, which is a vascular layer, and that includes the choroid in the back, which can be seen here in this area, right from here to here. And if you follow it around towards the anterior segment, it includes the ciliary body and its processes, the iris, and the lens, which is pretty blown out here. The whole center has failed to be properly fixed and embedded and sectioned and so on. The innermost layer really only exists at the back of the eye, and that's the retina. And we are going to examine the retina in some detail a little bit later, but just for now it's all of this layer here, the innermost. So I said earlier that there's some features on this slide that you can't see on the other ones, and that includes these sections of the eyelids here and here, and that will show you the conjunctiva, which is the inner surface of the eyelid here on this side, and it reflects back onto the eye itself here, and then transitions into the surface of the cornea over here. So let's look at that at a little bit higher magnification, and you can see that it's very different from the skin side of the eyelid. It is a wet membrane, a mucous membrane, so the conjunctiva would be here and here, both the inner surface of the eyelid and on the eyeball itself, covering the sclera, which is down here. We'll look at the eyelid again later on in histology when we look at skin, because we're going to look at some of the things that you see in skin and some of the glands that are present here. But for now, we'll just, mention the conjunctiva and let you know that that's what this structure is, the eyelid. Moving on to the anterior segment slide. This one really allows us to look in some detail at some of the structures in the front of the eye. And you do have a little bit of conjunctiva here as well, but you don't have the eyelid. That was removed from both sides here and here. So let's start at the outside and work our way in. Let's start by looking at the cornea. Recall that we've looked at the cornea a few times in the past when we looked at dense regular connective tissue, which is the stroma of the cornea, and we looked at the two epithelial layers of the cornea. And all of that happened way back in the beginning of the academic year, so it might be a little hazy. So let's just take a look at them again. The outer surface of the cornea is a stratified squamous, non-keratinized epithelia. You can see the flattened cells that are facing air. Out here is air. And underneath it is a very thick basement membrane that doesn't show up real well in this sort of prep. If we had a periodic acid shift one, this would be a bright pink stripe underneath it. And that's Bowman's membrane. It's a very thick basement membrane. The other side, which is not in focus right here, let's find a spot in focus. Here we go. The other side, the other epithelia that faces aqueous humor, is the corneal endothelium, and that is also underlain by a fairly thick basement membrane, which again doesn't show up really well in this particular prep. We could go up in magnification and try to distinguish it. Here we are, and this area right here where the hand is showing the basement membrane underneath this epithelial sheet, which is extremely thin. And that is Descement's membrane. So the way I like to remember those two names is alphabetically going from outer to inner. So Bowman's, Descement's. It's just my simple way of figuring that out. So in between them, of course, is this corneal stroma. And you notice that all of this material appears to be in one orientation, the cells and all of this matrix, which is mostly collagen fibrils. So what's going on here is that this is all aligned very precisely, and it's kept at a very specific hydration level, and that's controlled by this endothelial cell population here, the corneal endothelium, and they pump fluid so that this stays at the very constant hydration, and that is what makes this clear. So the interesting thing about the cornea is that it is actually the major refractive element of the eye. Most of the light refraction happens here. Most people think it's the lens, but the lens actually does less refracting, but it's in charge of variable focusing. So this can change shape, this cannot. Except, of course, if you get LASIK, in which course they change the shape of your cornea. So let's move on to the point where the cornea here meets the sclera. That's called the limbus, this area here. And at the inner surface where the iris base comes down and meets the sclera and the ciliary body, there's a very special region called the angle, the corneal iridial angle. And it has these kind of meshwork fibers, which unfortunately are out of focus on this side. So let's buzz over to the other side. Here we go. This example of the trabecular meshwork on the other side of this specimen is much more in focus, so we'll take a look at this. So this is where the aqueous humor drains into, goes into these channels, and eventually ends up in this little space called the canal of Schlemm. And from there it goes into the venous circulation. So the aqueous is produced back here by these cells on the ciliary processes, the outer cell population here, and it flows from the posterior chamber this way, out through the pupillary margin here, into this anterior chamber, and then outward here via the angle. So what else can we see here in this area? We can see some of the structures of the iris, and I'm going to jump back to the other side because of this piece of lens specimen that is artifactually sitting on top of this iris here. This one is in better shape. So looking at this, you can see that it has this very thick epithelial layer that is highly pigmented in the back, but that's not what controls the eye color. It's the amount of pigment in the actual stroma here. So if you have a lot of pigment in the stroma portion of the iris here, your eye is darker. If you have less, it's lighter. So what is the functional part of this? It's bands of smooth muscle. So here we have a band of smooth muscle that is very close to the pupillary margin here, right here, and we can get higher yet and ensure that this is indeed smooth muscle from its pink color and the way the cells are coming right at us. It's a sphincter muscle essentially, and when this contracts, it closes the pupil. How does the pupil open? Well, there are radially aligned bands of smooth muscle that are associated with the second epithelial layer right here. And you see this kind of pinkish layer? These are myoepithelial cells right here. And when these contract outward, it pulls the pupil back open. So this is the dilator, and this is the constrictor. Now let's take a look at the ciliary body. The bulk of the muscle of the ciliary body is in this region right here. There is some that stretches back in this direction that can help pull this backwards a little bit, but most of it is right here and is circumferentially oriented so that when this muscle here contracts, it closes the annulus, because this is a circular annulus muscle, just like the pupillary sphincter muscle. And these ciliary processes, which are attached to these zonules, which you can see little pieces of here, then move inward, and the tension on these, which is holding the lens in a flattened conformation, is released. So the lens rounds up, and that process is called accommodation. So normally the lens is held in a disaccommodated state, flattened, and then when the ciliary muscle here contracts, it rounds up and allows you to see near vision. So it increases the focusing power of the lens. The lens itself is very difficult to fix properly, and large lenses are really just almost impossible to get much of except for the outer ring like you see here. But you can see a couple of interesting features. Number one, you can see that the bulk of the lens is made up of these long skinny cells called fibers. You see how long and thin they appear. And they are void of all organelles, and that is one of the characteristics that promotes the transparency of the lens. There is only a thin epithelial layer at the anterior side, and the whole lens is enveloped by a very thick basement membrane, the lens capsule. New lens fibers are made here at the equator of the lens. They differentiate from these epithelial cells, and they begin to elongate. And then as they begin to differentiate, they lose their organelles and they become just bags of special protein called crystallins. And it's both the molecular arrangement of the crystallins and the very precise crystalline arrangement of the cells themselves that promote that transparency. So the lens can undergo changes with age. It can become opaque in the center, although you can't see the center here, and that's very typical with aging. It also stiffens up with aging and does not focus at near distances very well, so it doesn't accommodate well in older individuals. This is called presbyopia. Next, we're going to examine the posterior segment structures. Let's start by looking at the posterior eye segment with macula fovea slide. So let's zoom on up here a bit and take a look at this structure. So beginning with the basics, let's identify first the sclera on the outside, the choroid, and then the retina here, all of these layers. You can see right off, even at this kind of lower magnification, that the retina is made up of several cell layers, which we'll explain in detail in a couple of minutes. So let's go up in magnification a little bit further and center this thing. There we go. So let's start at the inner side with this inner limiting membrane, and then this would be the actual fiber layer, and then this would be the ganglion cells. So the fiber layer is the axons of the ganglion cells. Then you have what we call the inner plexiform layer and the inner nuclear layer, then the outer plexiform layer and the outer nuclear layer. Then right about here, it's not incredibly clear in this particular image, you have what's called the outer limiting membrane. Then you have the rod outer segments, rod and cones, and all of that is called the neural retina. The last layer, which is this thin one here, is the retinal pigment epithelium, and that's the final and outermost layer of what's called retina. So what's in these things? Well, I said this is ganglion and this is axons. Here is the interface between all of these cell types and the ganglion cells. So it's where they are synapsing and exchanging information and so on. There are 4 different cell types in this inner nuclear layer, and those are amacrine cells, bipolar cells, horizontal cells, and Mueller cells. Now, with this sort of histological preparation, you cannot really distinguish those cells from one another, so you won't be asked to do that. However, you will have to know what cells are present there and what their functions are, which is all in your self-study material. So moving along, this inner plexiform layer is where these cells interface with these cells here in the outer nuclear layer, and these are all the nuclei of the actual photoreceptors, in other words, rods and cones. They have what we call the inner segment, which is the nucleus and most of their cytoplasm, and then the cytoplasm of those Mueller cells ends right here at that outer limiting membrane. So all of this stuff is covered or kind of encased in Mueller cell cytoplasm. However, that ends, and all of these outer segments of the photoreceptors are not covered by the Mueller cell cytoplasm. As a matter of fact, they interface towards their tips here with the retinal pigment epithelium. So this is where light coming in goes through all of these layers and strikes the photoreceptors here, and then the signal is propagated and processed by all of these cells and sent back out that way towards the optic nerve. So retinal pigment epithelium here has a lot of functions, as talked about in the self-study material. And you can see that it has pigment in it, along with the choroid, which is heavily pigmented. And all this pigment here helps to absorb the reflections and minimize glare of light that makes it all the way through past the photoreceptors, so it doesn't bounce back and cause a lot of problems. The choroid out here is actually very heavily vascularized. It's the blood or source of oxygen and nutrients for all of the outer retina, all of this stuff here. There's vasculature here, which sometimes feeds this inner layer, but most of the source of the blood is here in the choroid. Now let's look at a very specialized region. It's going the wrong way. There we go. Now let's look at a specialized region called the fovea. So the macula is the macula lutea, and that is this larger kind of area around here. And that can't be really distinguished histologically, but usually it's distinguished on examination looking into the eye with an ophthalmoscope. But histologically, you can see this depression kind of area here, where these outer nuclear layers are kind of minimized or pushed to the side. That way light can come in and can strike these photoreceptors with very little interference from anything else. So here is where you have a high concentration of cone receptors, and you have high acuity vision here. So let's look at that at higher magnification. We'll just zoom right up there, and you can see the outer receptors. Now at high mag, you can see that outer limiting membrane right there. And here's the photoreceptors. And then they're interfacing here with this retinal pigment epithelium. Underlying that, there's this pink kind of line. That's an example of Bruch's membrane, which is the membrane that separates the retina from the choroid. The final slide we're going to look at is the slide of the posterior eye segment that contains the optic disc. So the optic disc can be seen as a disc only when you're looking into the eye. From this orientation, you're looking at kind of a cross section through the disc, which is right here. So let's go up in magnification and look at some of those characteristics that it has. So the first thing you're going to notice about this, even at a lower magnification, is that there's no layers. There's no nuclear layers, there's no plexiform layers, and there's, most importantly, no photoreceptors. So this is a blind spot. There's no actual image production that can occur here. However, this is where all of the fibers from the ganglion cell layer go to. So this is their point of exit from the eye. They all come to this point and they exit out into the optic nerve, which is here. And they do that through this collagen kind of network called the lamina cribosa. And the significance of that is that just after they exit that lamina cribosa area, they become myelinated. So they're unmyelinated here and they're myelinated out here. So this is pretty nice because you can actually get another look at the retina here if you did not get enough details from the fovea slide. So you can look at this pretty nicely at higher magnification, and you can see all of those nuclear layers and plexiform layers that we were examining earlier. And this is a real nice example of the retinal pigment epithelium too, and the choroid underlying it. So that's all we have for today. As always, if you have any questions or concerns, you can send me an email, and we're going to have, towards the end of the block, a short review session on Zoom, most likely.